So for a little bit of background, this session is going to span the gamut from basic science like creatine synthesis at the cell level to behavioral effects in mice models for CTD, and finally back to potential treatment. And the treatments are, again, a big theme for the day has been gene therapy, and you're going to see that reflected in the keynote talk too. But I want to emphasize that the potential treatment in this particular session is gene therapy for Gamt research. So we're going to come back to that talk. I would like to first introduce Dr. Stephen Baker, who's coming from University of Utah more recently, but was previously affiliated with Stanford School of Medicine. And we haven't met Stephen actually, so I'm very excited to get to meet face to face. As you folks know, the ACD kind of trolls PubMed periodically for any papers that may mention any of our genes or any research that is relevant. And this is how we got to meet Steve and invite him to present at our conference. And I'm very excited that I've seen lots of questions from you, Steve. So I'm sure we're going to keep talking.
Yeah,
so take it away, Steve.
Very much so. Thanks, Laura. Yeah, it's truly an honor to be invited to speak here. So, and I'm humbled by my co-presenters here. So thank you so much. So today I'm going to talk to you a little bit about how creatine is synthesized in the body and particularly in the brain. To do that, we wanted to really map the two synthetic enzymes, AGAT and GAMT, using immunofluorescence. And in order to start that experiment, we relied on the findings of others and some single cell RNA sequencing data. So I'll show you that first. So of course, people in the field are very familiar, and one of my personal heroes is Olivier, who's here today, who published this paper in 2001, which really broke ground on the idea that these synthetic enzymes, so I'll call them today GATM or GATM and GAMT. So GATM you probably know best as AGAT, but at the gene level it's called GATM. So this groundbreaking paper in 2001 showed that these two synthetic enzymes, as well as the creatine transporter, are expressed throughout the brain in a number of cell types. And what we wanted to do was take a closer look at this foundational work using this new technology of single cell RNA sequencing that has become publicly available in these large consortium data sets. And so what we did was we wanted to ask the question, where do we see these genes expressed in the brain? And what is the hierarchy of cells that are expressing them quantitatively? And then where also do we see them expressed throughout the body at single cell resolution? And so we harnessed the data that was made available by the Tabula Muris consortium, which essentially took a number of mice, broke those mice down into single cells, and then sequenced their RNA to ask where are these two enzymes. So they, of course, surveyed all 20,000 genes approximately in the genome, but we wanted to look at the two synthetic enzymes in the creatine pathway. And so when we do that, if you aggregate the profiles for GATM at the tissue level, what we were surprised by was that the brain actually shows the highest median level expression of GATM, even higher than the kidney, where it's canonically thought to be expressed at high levels. And if you look within the brain, what you find is that actually oligodendrocytes and oligodendrocyte precursor cells, so these are just showing you the density, where very high levels here are expressed in that lineage, so in the oligodendrocyte lineage, even higher than any other cell type in the brain. And the second highest cell type that expresses the first synthetic enzyme is actually endothelial cells. Whereas if you look at GAMT, GAMT is, of course, highly expressed in the liver, which was expected, but we also detected a significant amount of expression in the brain. And if you drill down and ask what cell types are expressing GAMT, again, you find that oligodendrocytes sort of reign supreme in terms of their expression of this second synthetic enzyme, followed closely by parasites, so these perivascular supporting cells. And so we took a survey of all cell types across the entire mouse body. And what we were very, very surprised to find was that oligodendrocytes not only express the highest level of each synthetic enzyme, but they also highly overlap within that particular cell type. So the size of the circle indicates a very simple metric of cellular overlap, which is called the Jacquard index. And so bigger circles mean that the same cell types are expressing both enzymes. And so really oligodendrocytes expressed more GATM than any other cell, including cells of the proximal convoluted tubule in the kidney, and more Gamt than even hepatocytes in the liver. So we were very surprised by that. And it said that oligodendrocytes might play some very special role in expressing or generating creatine. And in order to sort of study this at the immunofluorescence level, we wanted to start with a model tissue that was well characterized. And so if you look here in blue, what we're showing you is the liver, of course, expresses high levels of Gamt in the hepatocytes, and that was known due to previous characterization. But we also found high levels of GATM in the endothelial cells of the liver. And so we took a look at these RNA expression patterns at the protein level using immunofluorescence. And here I'm just showing you an image of the liver, and blue is showing you the nuclei. Albumin is showing you where the hepatocyte cytoplasm is. And then we use an podocolixin, which is an endothelial cell marker, to stain the endothelium. What we found was that Gamt is, of course, highly expressed inside the hepatocytes, but GATM shows these discrete puncta, which are right adjacent to nuclei of the endothelial cells. And so there does seem to be quite a bit of expression of the first synthetic enzyme in the liver as well. And so this is just showing you a three-dimensional image of that same sort of field. And so what you can appreciate is that if you're looking at blue for nuclei, albumin is showing you the cytoplasm of the liver cells, the hepatocytes, and GATM here is showing you these flashing puncta that are adjacent to these nuclei within the sinusoidal endothelium. And here we just overlay the endothelial marker to show you that they're co-localizing with that endothelium. And surprisingly, GATM was within the endothelium of the sinusoids, but in here in this large central vein, we actually didn't detect any GATM, and we also didn't detect any GATM in the portal vasculature as well, suggesting that this sinusoidal endothelium has a special property of expressing GATM, and it may form a functional circuit where it produces GAA and then transports it into the hepatocyte, which is then expressing high levels of Gamt to convert it into creatine. And so how about in the brain? And so for our tissue, subcellular or sub-tissue model system in the brain, what we used was the cerebellum, because it has this nice laminar structure where you have a segregation of neurons from glial cells or oligodendrocytes and astrocytes in the white matter tracts. And so here I'm just showing you a 3D volume that was taken by confocal microscopy and then just rotated so you can see the contours of the cells. GATM is the first synthetic enzyme, so AGAT is this red signal that's around all of these nuclei that are in the cerebellar white matter. And then the green stain here is for nu-N, so it's showing you neurons. And so here you can see the granule layer of the cerebellum. And all of those cells are negative for GATM. There are actually some cells that are tucked in here, which are nu-N negative. And I'll show you on the next slide, those are actually oligodendrocytes. But so all of these cells here in the cerebellar white matter are where you find the GATM expression. And so we stained for Olig2, which is a lineage marker for oligodendrocytes and OPCs. And what we found here is you can see here in the cerebellar white matter, there are lots of Olig2 positive nuclei, which are surrounded by these GATM foci. And here up in the left-hand corner, you can actually appreciate there are some Olig2 positive nuclei that do have those GATM foci around their nucleus, so within the cell body. And so we quantified across three different areas of the brain, the cerebellar white matter, the corpus callosum, and also the cerebral cortex. Where do we see expression of what cell types do we see expression of GATM in? And we used a number of lineage markers. So we used nu-N for neurons, Olig2 for oligodendrocytes, GFAP for astrocytes, podocolixin for perivascular cells, and then CD68 for microglia. And in all of these cell types, what you find is that really the dominant cell type that is expressing GATM inside the brain is oligodendrocytes, accounting for about 90 to 95% of all cells expressing the first synthetic enzyme. So what were these puncta that we were visualizing around the nuclei? It had been previously shown that GATM localizes into the inner membrane space of the mitochondria, so a compartment between the matrix and the outer membrane. And what we used was a marker here for the inner membrane of the mitochondria, so COX-1, which is a member of the electron transport chain. And what you find is that these puncta of GATM or AGAT are co-localized very nicely with this mitochondrial marker, suggesting that around these, inside these oligodendrocytes, GATM is localizing to the mitochondria, which is where we'd expect it to be. And so what about the second synthetic enzyme, GAMT? So here is another three-dimensional volume of the cerebellar white matter. And again, staining for nu N to show you where the granule layers are. And so what you can see is here in the white matter tract itself, you can see a number of cells that are positive for GAMT. And it's actually almost like a bimodal appearance where you have these sort of dimmer cells and then these very, very bright cells that are positive for GAMT. And when we stain this area for Olig-2, of course, these cells in the center here are positive for the green stain, indicating that they're oligodendrocytes, even the very bright ones. And so by far and large, the cells that are expressing the second synthetic enzyme GAMT are oligodendrocytes inside the white matter tracts. And so our last question was, does this extend to humans? And so in order to address this question, we collaborated with a former lab member who it was from the Montine lab at Stanford, who's now moved on to UCSF. And what he did was he did single cell attack-seq using human brain. So from 4 different regions of the human brain, so the cortex, the hippocampus, the striatum, and the substantia nigra. And he asked what regions around the chromatin of GATM and GAMT are accessible inside the brain of humans. And so what we found was that what you typically expect is most genes, their promoter is very accessible, and that's what we found here for GATM. But interestingly, there was this very accessible putative regulatory element within intron 2 of GATM, and it was the most accessible in the oligodendrocyte lineage and also in oligodendrocyte precursors with lower levels of accessibility in astrocytes. And then as far as GAMT, what we found is, of course, as we'd expect for most genes, the promoter was accessible in GAMT across cell lineages, but there was these 4 very active putative regulatory enhancers within 16 kb of the promoter of the GAMT gene in the human brain. And these were specific for oligodendrocytes. So in conclusion, GAMT or GATM and GAMT mRNAs are highly expressed in oligodendrocytes more than any other cell type in the body. And local protein expression of GATM and GAMT can be found in multiple organs. So here I showed you the liver and brain, but we also have data for the kidney, testes, and heart, where both synthetic enzymes are expressed. And in the brain, oligodendrocytes comprise the majority of cells that express GATM, AGAT, and GAMT. And this is particularly, these are particularly dominant cell types in the white matter tracts. The first synthetic enzyme, GATM, localizes to the mitochondria. The second synthetic enzyme localizes throughout the nucleus and cytoplasm, which is consistent with its small size and permeability to the nuclear membrane. And in the human brain, oligodendrocytes and OPCs exhibit a very highly accessible enhancer within intron 2 of GATM. And so one thing that we're very interested in is, can that enhancer be harnessed, for example, inside neurons to turn on the creatine synthetic pathway in the cell types that need creatine? So we were very excited by that finding. And so I'd like to thank my collaborators on this work. So in particular, the Montine lab. And then also we got some really helpful advice and a collaboration from Dr. Isbrandt and his postdoc Malti at the DZNE. And then our collaborator, Ryan Corses at UCSF, and our funders, of course. So thank you.
Thanks so much, Stephen. This was great. I think, you know, you're a welcome member of our community. So I hope that we see a lot more of your research in the upcoming months. I would like to invite Dr. Skelton, and Matt is a longstanding member of the ACD family, and he's part of the Scientific and Medical Advisory Board. And fortuitously, we are asking Matt, what do we know about ADHD in CTD patients? And Matt came back and said, well, I have exactly the results to show you. And I think he's going to share it with all of us from mice data. So take it away, Matt.
Thank you. Okay. And everybody sees the title slide?
We do not. Okay, now we're seeing your screen. We're seeing your monitor, and I see your PowerPoint bar there. There we go. Now we're seeing it. Great.
Great. So yeah, so today I want to talk about looking at executive function in our creatine transporter knockout mice. So a brief overview of what is executive function. These are the processes that control information processing. So it's basically how do you follow the rules. And this includes things like attention, working memory. Working memory is the ability to remember a short sequence of words, numbers, etc. For a short period of time. So sort of the classic is remembering a phone number that somebody tells you. Back I had to remember phone numbers. Inhibitory control, planning, monitoring, things like that, cognitive flexibility. As Lars said, ADHD is a frequent comorbidity with creatine transporter deficiency, and we wanted to examine this. So why is it important to test executive function? And I think that Dr. Curie did a good job of this as well, explaining this earlier, sort of a serendipitous prelude to my talk. But it addresses the root causes of some of the behavioral deficits that we see. So we've kind of gotten into a rut where we're looking at the same 3 or 4 behaviors over and over and over again. And one of the critiques we were getting is you need to look at other things. You know, we agreed. The other advantage is that the tasks, especially the last one I show you, will better match tests that are used in humans. So you'll see some overlap between what Dr. Curie presented and what I'm going to show with the 5-choice. You know, when we test cognition in animals, we ask them to swim, we shock them, we ask them to look at 2 objects and tell us which, you know, and spend more time with one. And humans aren't asked to do that during a normal cognitive battery. These tasks are also dependent on physical performance, which is also an issue, especially with the ubiquitous knockout mice who are smaller than their wild-type counterparts. In addition, it just provides us with a greater depth of knowledge. And I think that's important as we start to really get into, and you can see through some of these talks, the difficulties we have in designing treatments. Seems like a very simple problem, but it indeed has become a very complex issue. And I think the more we know and the more outputs that we have, the better chance we have of developing a truly translational, relevant model to give a high degree of confidence for any treatments that we find. Lastly, the pathways involved in many of these are well understood, mostly in the prefrontal cortex and the limbic system. So, you know, these are well mapped out, so we can then use the findings that I'm going to show today and sort of drill deeper into this and really start looking at pathways that could be affected. Again, that gives us maybe a chance of alternative treatments and things like that. So how do we test this in rodents? There's a variety of ways to test executive function. I'm going to talk about 3 today, but real quickly. You can test working memory in a special version of the Muris maze. You can use the Y maze, which I think most of us are fairly familiar with. The Radial Arm Maze, delay match to sample. Attention is attention set shifting, which we won't talk about. In the 5-choice cell reaction time test. Inhibitory control or things like the continuous performance task. And then cognitive flexibility is delay non-match to sample, so you have to pick the one that's not like the others, so to speak. And then reversal learning in the 5-choice. So we picked 3 to look at executive function in our mice: the Y maze, the Radial Arm Maze, and the 5-choice. So first, we'll talk about the Radial Arm Maze. So we actually use a water maze version of this. It's an 8-arm maze. Here, we start the mouse in 1 arm, and all the other 7 arms have platforms that they can escape. So trial 1, they'll swim around, they'll find a platform. So when we take the mouse out of the maze, we take the platform with it. That arm is no longer baited. The mouse has to remember not to go back to that arm because there's no escape. So as they go through, and then so we do that for 7 trials. Each time they find a platform, we take it out. So by trial 7, there should only be 1 platform left if the animal is performing the task pretty well. So here are the data that we do this over 2 days. And here are the data that we have. We use the ubiquitous knockout mice in this case. We measure latency, so that's just the time to find the platform, and we measure errors, and that's entry into an arm that's already been found. There was no difference in latency between the wild type and the knockout mice. There was a small increase or small decrease in the number of errors committed. But interestingly, when we actually took and looked at animals that failed to find the platform, and we assigned a maximum error score, and I didn't show this, but we assigned a maximum error score to those mice. When we did that, it eliminated those deficits. So while they show that decrease in errors, it could be a decrease in exploratory activity, which again goes back to the original thing of when we ask an animal to physically perform, that may not be able to physically perform as well. This is the kind of confounds that we run into. So the next thing that we looked at was the Y-maze. And this is a test of working memory. We did two versions of this. We did a spontaneous alternation, where the animals just put in one arm and allowed to move around. Ideally, it'll remember where it visited before. So it goes to the start, he'll come here. When they come back out, they're gonna go this way. And when they come back out, they're gonna go this way. And that's counted as a spontaneous alternation. We count the total number of entries and we count the number of times that it goes from A to B to C or any combination of that. So as long as it doesn't re-enter an arm twice. So there's no difference between our mice. So I know that Laura's group has shown conclusively that they have Y-maze deficits, spontaneous alternation deficits in their mice. We weren't able to replicate that in ours. It's sort of an interesting difference between the two and kind of highlights why we need multiple models as well and show that these treatments work across different type of models because the kids are different too. The patients are different as far as manifest. Interestingly, we did get a trend towards a lower number of total entries, but that didn't quite clip significance, which would suggest the animals may have some anxiety or maybe hypoactive. We'd have to follow up on that with total distance travel. Next, we did forced alternation. And in this task, we block this arm and we let the animal just kind of walk back and forth here for about 8 minutes. We wait an hour, we remove the partition, and then let the animal explore both. Ideally, he's going to spend more time in this arm. There's no difference between our wild types and our knockouts. This is the group that you would be looking at, the blocked. And interesting, we didn't see a strong preference, and we analyzed this by time as well. We didn't see a strong preference for this arm. And this does highlight some of the weaknesses of the Y-maze. While it's a good test and it's powerful, at times you just don't get the right conditions to really get a nice, robust output from it. So we'd like to actually try to modify this a little bit by changing the light levels and changing our cues and stuff like that in the room and see if we can hopefully replicate ours findings. So the next test we went to was the 5-choice cell reaction time test. So because we food restrict animals, we use the brain-specific knockout mice in this case that we had developed before and published. We food restrict them for about a week before. We get them down to about 85% of their body weight. Nothing too significant, but just to make sure that they're hungry when they go in. So mice are required to nose poke into a hole that's illuminated. So on one side of the chamber, there are 5 holes. On the other side of the chamber, there's 1. This is the reward hole that we'll deliver the food through, the strawberry milk through. So when the trial starts, hopefully it will go through here. The light comes on, the animal pokes his nose, the light comes on in this hole, and he's given a strawberry milk reward. Mice love strawberry milk. It's a heck of a reinforcement for picking the right hole. If the animal makes a mistake, there are 3 types of mistakes the animal can make. The first mistake is an incorrect selection. So the light comes on here, he decides to go over there. When that happens, we turn the lights off on him and he doesn't get a reward. The second type of mistake is an omission. So the light comes on, the animal does nothing for a period of time. So the stimulus duration is one of the main factors in this. So we start with a 10-second stimulus duration. We work our way down to a 2-second. So if he doesn't actually do anything for that time, the light gets turned off, he gets nothing. The last is a premature response. So after he takes his reward, light goes off, the dipper drops, and he runs over and pokes his nose into a hole before a light comes on. We turn the lights off and he gets nothing. So he gets each mouse gets 120 trials a day. It takes about 6 to 8 weeks to run this test per mouse. We have a couple of chambers, so it's not too bad. one of the main things we have to do is train the animal in sort of the rules of the test. So again, I mentioned the stimulus duration, the amount of time the light is on. So after a brief introduction where we show them that the reward hole gives them food, we turn around and start randomly selecting a light and presenting it for a certain amount of time. We start with 10 seconds, we work our way down to 2. They have to reach criterion to pass from 10 seconds to 8 seconds, and then 8 seconds to 4, and then 2. It's the number of responses and a certain amount of response time. So you can see that during the training phases, the animals do pretty well at this. So accuracy is just correct versus incorrect. We don't count omissions in accuracy. There's no difference between groups during the training phase when it comes to accuracy. So they're picking the right hole for the most part by the time they've gotten trained. Interestingly, though, the brain-specific knockouts do show a slightly higher level of omission. So they're just not paying attention to the lights as much. And there's no effective time on this at this point. So there's no real, it's just a general inattention. So then we also measure days to criterion, and there's no difference between groups in that. And the minimum amount of time that the animal can spend at one stage is 2 days. And this does include, I should also mention, this does include all the control groups that are required to make our brain-specific knockouts. So we have wild-type animals, we have our floxed animals, and we have our Crea-expressing animals. So there's no difference between those 3 groups, so they're consolidated as a control group. So once they've gone through training, we start to mix it up on them a little bit. The first thing that we do is we change the stimulus duration. So we give them a random stimulus duration. So it's anywhere from 0.2 to 1.6 seconds. There are 4 different ones, so blocks of 30, and they're selected randomly. What we see is a main effective gene for accuracy. So the animals aren't as accurate in their selections. They make more incorrect choices. Their response time is similar. So the processing speed seems to be similar between groups. They do commit more omissions during this time. So the inattentiveness is there. But they do not commit more premature responses. So premature responses are generally thought as impulsivity. So then after we do that for a few days, we go to the next phase of testing where we change the intertrial interval. So this requires the animal to be patient. Usually, so during all the previous training and testing, the intertrial interval was actually 4 seconds. So now we change it from 3 to 7. So obviously you would think that at that 7 seconds, they're starting to get a little antsy, like, okay, where's the next one? Animals that have impulse control problems will show a high level of premature responses compared with wild types. Interestingly, our knockout animals show less premature responses. So it should suggest that they do not have an impulsive type of deficit here. But they do, again, show an increase in these omissions, which is a sign of inattentiveness. And then perseverant responses are just, they go back to the same hole that they got the reward from the previous trial, even if it's not lit up. So they just, if they get hole 2 and they get a reward, they go immediately back over to hole 2 and poke their nose in, which is obviously a mistake and a sign of repetitive type of behaviors. Okay, so summary here is that we did have a reduced errors in the Radial Water Maze, which is a test of working memory. This may be due to less exploration, so it might be beneficial to send our brain specifics through to sort of mitigate the physical deficits that they could have. No differences in the Y maze, and then our brain specific mice show deficits in the 5-choice cell reaction time test. Decrease in accuracy, which is consistent with attention deficit models of attention deficit hyperactivity disorder. Increased omissions. So one of the things I didn't mention was that ADHD also, has there's several different types of ADHD. One of them is inattentiveness, and the other tends to be the impulsivity type. And it appears that ours have the increase in attentiveness type, is that they just don't pay attention to the lights coming on in time to make a correct decision. Decreased premature responses, which would suggest less impulsivity, but it could also suggest that they maybe don't like the reward as much. I doubt that, because when we look at the number of rewards they take and their latency to go get the reward, there's no difference between the two. And then no change in latency to a correct response, which again, that goes to processing speed. So when you're talking about things like the Wisconsin card sorting task or delayed match to sample in humans, those are important measures as well. And then future directions, I'd love to do the continuous performance task. This is basically a go, no-go type of task. Delay match to sample to better assess working memory to get a little bit more of a direct. So in that one, we shine one light, they get a reward, they go to the reward hole, they don't get a reward. We shine two lights and they have to pick the light that they picked the first time. So they have to remember that first hole. And then progressive ratio, which we increase the number of hole pokes they have to do each time. And that gives us a measure of motivation. And then finally, of course, we want to try to start to elucidate mechanisms that underlie these deficits. So we're going to look at things like dopamine specific knockouts, or looking at dread, looking at activating parts of the prefrontal cortex to see if we can either mitigate or exacerbate these deficits. And the acknowledgements that everybody in the lab, our animal behavioral core, division of neurology and all funding, especially the Association for Creatine Deficiencies. With that, thank you for your attention.
Thanks so much, Matt. So anecdotally, and from my kids' experience, I can relate to the increased inattentiveness model for ADHD rather than lack of impulsivity or too much impulsivity. So Mikelle and Olivier, feel free to take turns and ask all of our speaker questions. I think we have a good starting point in the Q&A. We have about 30 minutes.
Perfect, thank you. This question is for Dr. Skelton. What are medications for ADHD that work for inattentive models of ADHD? Could we test out if CTD mice improve with particular ADHD drugs? Arguably, a large number of CTD patients take ADHD medications, and it's never clear what would work better for our specific phenotype.
Yeah, great question. Yes, we can test it. So as far as, I don't know what medication would actually work better for that. The thing about ADHD is there's a lot of trial and error. There's a lot of, what works best for each individual patient. And a lot of that has to do with
Matt, we're losing your audio a little bit, so maybe keep that. Yeah, thank you.
Okay. Yeah, so some of that is just, what other sort of, gene drug interactions you might have. So, what works for one person doesn't work for another. But yes, the 5-choice is a really great way to test that, because once they've gone through the training phase, we can give them drugs that actually, we can give them amphetamine, give them methylphenidate, and test how those changes, how they respond differently to them, and see if it decreases the amount of omissions and incorrect responses that they make.
Thank you.
So maybe I have a question for Steve. First, it made me feel old to see my very old mRNA labeling at the start of your talk, dating back to 20 years ago. My question is, what do oligos do with all this creatine they are supposed to synthesize?
Yeah, that's a great question. So there is some really excellent data that oligodendrocytes, at least during myelination, and particularly during differentiation in culture, show reduced apoptosis when they have a functioning creatine synthesis or are supplemented with creatine. So oligodendrocytes are probably one of the end users of the creatine that they're synthesizing. However, it's really compelling to think that they are producing it as sort of factories for other cells within the CNS, and particularly neurons. So neurons, if you look at the single cell RNA-seq data for CT1 or SLC6A8, you do see expression in oligodendrocytes, and you see it in neurons. And so that suggests that those cell types are taking up creatine from the CSF. And it's hard to imagine if it's possible for oligodendrocytes to synthesize the creatine and then export it if they're also expressing CT1, but it could be positional. So within the myelin sheath, there could be expression of CT1 on the outside, and then on the inner myelin sheath, when it's in contact with the axon itself, there's efflux channels. So that's something that's tempting, but we certainly haven't shown that yet. So that's just speculation.
And to continue on this, what in case of a creatine transporter deficiency?
So what was the question, Olivier?
Within the oligos and this huge expression of AGAT and Gamt. Do you think there could be some regulations or no effect in oligos?
Absolutely. Oh yeah, that's a great question. So it's been shown in peripheral tissues that in SLC6A8 knockout animals that AGAT expression increases and GAMT expression decreases. It depends on the tissue type. I think the effect was significant in the liver for GAMT, but didn't reach significance in the muscle. But certainly there is some negative feedback on AGAT levels. And I think one of the talks actually today was excellent about that idea. And so there's probably regulation in the absence of loss of CT1. And that's something, since there are multiple, at least 2 CT1 knockout animals, one made by Matt, that we'd love to study and see, what the expression is in oligodendrocytes and if it's modulated in that setting. That's a really great question.
Okay, thanks.
The next question is for Dr. Lipschutz. Dr. Lipschutz, do you think the age of the mouse matters for extent of recovery? Meaning whether the gene therapy is administered to wean-age mice or older mice? And relating to humans, what would you expect to be rescued in older individuals with Gamt with gene therapy?
Yeah, that's a great question. And there's a number of questions also in the chat for me that you might have seen. But to answer your question specifically, so this is a little bit of a challenge. If we I know that if we administer this vector to neonatal mice, which are literally this big, they're about 1 gram to 1.5 grams at birth. And if you consider an adult mouse at 25 grams, much of the expression will be lost in about 5 weeks. Now that same number, 5 weeks, won't apply to a human, but there will be decline over time. We are working on another approach that based on the CRISPR technology for integration of the expression cassette at a specific location to get over growth-based loss of expression. I would say that for for an adult patient or a larger child, this therapy has a much greater chance for early success because the liver has obtained near adult size already, since we're talking about liver-based expression. So that expression should be much more stable. There are studies out presently in the hemophilia clinical trials where adults were administered AAV who were out more than 10 years, continuing to express factor 9 hemophilia B. What could someone gain? Well, I think unfortunately a patient who already has deficits, I'm not sure that those deficits will improve. Certainly, it's hard to know. Certainly with oral creatine, I would imagine, this has been looked at, but pharmacologically, there's going to be peaks and valleys that may affect different cell types and different function as maybe oligodendrocytes or the cells, as Steve has just pointed out. But this will lead to stability. That is, once we have expression, the expression levels should remain high, at least for long term you would expect. Again, never been done before. The data is based on what we have in the hemophilia human trials, but I think this is what you would expect.
Thank you.
So reversal, I'm not sure that you would have, but maybe you could have stability or maybe some minor improvement. It's hard to know, I think. Do you want me to answer the other ones in the chat at this point, or do you want to direct those to me, or you tell me?
I think we'll continue to go around and hopefully we can get to all the questions.
I have also a question to Gary concerning the doses you use of AAV vectors. Did you observe other effects on the liver with the highest doses of AAV? you used, no special adverse effects?
We didn't see any adverse effects. Now, we at the 30-day mice, the dose finding mice, we didn't see any evidence of cell death or apoptosis in the hepatocytes at all. We have seen nothing like this. We've done other studies in our arginase models and in carbonyl phosphate synthetase where we've given this dose or higher and not seen effects also. So I don't think that we're missing anything here. I think before an invention, hopefully this could go to trial at some point, those types of studies would be done in detail, but we have not seen any pathologic effects in the liver.
Okay, I have a question for Dr. Baker. Can we promote creatine synthesis in brain cells with any other precursors? It seems that glycine and arginine in CTD patients did not work very well in the past.
Yeah, so I'm not a particular expert in the treatment of these deficiencies. So I'm a clinical pathologist. I do transfusion medicine, but I know that those are supplements. So yeah, giving the precursors, so glycine and arginine, can certainly promote the synthesis of guanidino acetate. So it's possible, and then that can be further converted into creatine. But I think it's one possibility that has been tried in clinical trials for patients with CT deficiency. And the data that am aware of are that it's not super effective in terms of crossing the blood-brain. Well, it may cross the blood-brain barrier, but may not promote the accumulation of creatine within the cells that need it. So it's certainly something that could be tried. One thing that we were excited about in our paper was that in intron 2 of the first synthetic enzyme of AGAT, essentially there's an enhancer that is specific for oligodendrocytes. And you could imagine if creatine is needed inside of neurons and that enhancer is turned off, one thing that you might do to promote creatine synthesis in neurons would be to activate that enhancer inside of neurons and have them supply their own creatine. There may be some sort of reason that evolution has chosen not to have that strategy happen, but it's certainly something that could be tried therapeutically. So yeah, it's a great question.
Thank you.
Maybe one question to Matt. How can we explain the differences between the different models of CTD?
Oh man, that's a big question. Some of it has to do with slightly different testing regimens between labs. For instance, things like the Muris maze, the size of the platform that you use in relation to the size of the pool that you put the animal in makes a difference. Like with us with the Y maze, I still feel like we need to optimize that a little bit more. We need to find sort of a nice balance between, if you have bright lights, the animal's going to be more nervous.
Take your microphone maybe nearer.
Oh, I'm sorry.
Yeah.
So like I said, a lot of it could be different testing regimens across labs. It might be a big part of it. Some of it may be the mouse background that they're on. So I do know that the original background for Dr. Brancelli's mouse is different than the background that mine is on. They're both C57 blacksticks mice, which for the general audience doesn't mean that much. But one of them was derived from one mouse repository and the other was derived from a different mouse repository. That makes a big difference, actually, because there was a mutation that occurred when Jackson sent those mice to Charles River that the C57s from the two different repositories are actually different than one another. That's a fairly significant problem that we need to reconcile. I think that, sort of a long-term thing that or sort of a pipe dream type of thing is that I would like to actually get a standardized testing regimen. I send mice to them, they send mice to me, and we just sort of see what happens. And the type of mutation might be an effect as well. There might be something in the mutation that we generated that's causing some secondary effects. There might be some sort of shift in other genes that are around the creatine transporter, because there are a lot of genes on the X chromosome that are very important for cognitive function. So, they could have disrupted something else. I think we've than RNA-seq. I haven't seen anything that's close to those gene, close to the creatine transport that's got differential expression in the brain, but that could be an issue too. As far as anything beyond that, I wouldn't even begin to speculate because I don't know, to be honest.
Okay, thank you. I have a question for Dr. Lipschutz. Will a Gamt gene therapy treated mouse also still need creatine supplementation or any other supplementation that they're taking today?
No, it won't. In the studies that, in fact, that we performed, we used a creatine-free chow. So they weren't, all the creatine that they have in the blood is either based from the diet, is based from the viral vector. There's none from the diet. We gave no supplementation at all. We specifically have a vegetarian chow with these mice.
Thank you. And when do you think the clinical trials will be available for human patients?
Well, that's an interesting question. I think it's partly dependent on the people that are in the audience here. That if they want something like this, because we didn't do all this work to have it not go anywhere. We did all this work because we saw the need in my conversations with Laura and Heidi quite early on. And so Heidi's been very supportive from the side of the society to obtain funding from the National Institutes of Health. We'll see what happens to move the work further forward. But I don't want to speak for Heidi and Sangeetha and Laura and Faith, but if you are interested in having a clinical trial, please mention that to them, because as part of the process, we or I need to know that there are patients or patients' families that want this, because that's going to be the argument with the FDA to file. So I would suggest strongly that you could feel free to reach out to me through email or phone call, but also please mention it to your board members, because I'm not in on everything that goes on the inside, but certainly this is where we're interested to go.
Thank you.
Thank you so much for that. And rest assured, we're definitely considering all possibilities, and we want to make it happen too. So we should get, take this conversation ahead and see where things can go. We are at the end of our Q&A session. Apologies, but I want to thank all of our speakers from the previous session, as well as our moderators. This is an amazing job.